My name is John Mascarenhas. I'm a professor of medicine at the Icahn School of Medicine at Mount Sinai in New York. So the study is a randomized placebo-controlled study in JAK inhibitor-naive myelofibrosis patients, randomized either to selinexor, 60 milligrams once weekly, plus ruxolitinib at the prescribing dose based on platelet count versus placebo plus ruxolitinib. And it had a co-primary endpoint of SVR35 at 24 weeks and absolute change in mean TSS score at 24 weeks. So at ASCO, we're going to present the primary data set at week 24, which shows benefit in the combination in terms of almost doubling spleen response with the combination, and then an equivalent reduction in total symptom score between the two arms. However, you do see an improvement in TSS with the combination from baseline. So although it was not different than the control arm, there was still this benefit that was quite obvious at 24 weeks. In addition, based on the fact that we saw this very rapid, deep, and sustained spleen volume response with the combination compared to single-agent ruxolitinib, we looked at survival. And with a median follow-up of 11 months, albeit with a limited number of events, we did see a signal that was encouraging and quite exciting to see for survival benefit with the combination. This survival benefit actually was also tied into SVR35. So SVR35 at 24 weeks, irrespective of treatment, was associated with an overall survival benefit. And this was seen in the combination versus single-agent ruxolitinib. Now, this is a median of 11 months of follow-up, so we're going to continue to follow the patients to see if this survival advantage continues to bear out. The safety profile was what one would expect with either agent alone and in combination, and mimicked what we saw in phase 1. I think with the addition of mandatory antiemetic prophylaxis in the first 2 cycles, it is very manageable to deal with the GI toxicity of nausea and diarrhea and alteration in taste. So this was not a major reason for discontinuation. In fact, got better with time. So the reality is, if you compare the 2 arms, there was really no difference in grade 3-4 treatment emergent adverse events between the 2 arms. Well, I think the company needs to meet with the FDA to review the data. It is a study that has a co-primary endpoint that was positive for SVR. It didn't meet this secondary co-primary endpoint of TSS. However, there was clear benefit in TSS score from baseline to week 24. And in my opinion, I don't know why we would need to beat single-agent ruxolitinib as long as it's consistent and not worse symptom improvement. In addition to the survival benefit, the reduction in spleen volume that correlated with survival benefit and reduction in driver VAF that was more frequent with the combination, I think with more follow-up, it is a study that I think does have an opportunity to go back to the FDA for consideration for approval. The other aspect is it is already an approved drug, selinexor, outside of myelofibrosis. So the question then becomes, since it's commercially available, is it possible to find a position in the NCCN guidelines with the caveat that that would be used off-label? Well, there's still more to come, so we're still going to be analyzing changes in bone marrow fibrosis and histopathology, reductions in driver mutation over time, survival, changes in cytokine expression, and then ultimately other non-driver mutation changes too. So there's still biomarkers that are pending that will also help us understand the aspect of disease modification, which again, I'll emphasize, was seen in a study here where spleen volume response, which was rapid, durable, with the combination was associated with a survival benefit, and that SVR did predict for survival benefit and associated with reduction in driver mutation. So all of these biomarkers suggest an on-target disease modulation effect of selinexor, an XPO1 inhibitor, with ruxolitinib. We'll see more data as it emerges with further follow-up.